Hello everyone and welcome to my channel Easy Life Science. Here I am talking about oxidase test and catalase test. So please subscribe my channel Easy Life Science. Like my video, share it to your friends and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. So let's start the oxidase test. The aim of oxidase test is to determine the ability of a microorganism to produce an enzyme oxidase. The oxidase enzyme plays an important role in an electron transport chain during aerobic respiration. Aerobic respiration is a respiration in which there is a molecule oxygen as a final electron acceptor. The enzyme cytochrome oxidase catalyzes the oxidation of reduced cytochrome by a molecule oxygen, and it results in a formation of a water molecule and a hydrogen peroxide. Aerobic as well as some facultative anaerobic organism and a microaerophilic bacteria shows a oxidase activity. In a cell or in an electron transport chain, the cytochrome C get reduced by accepting the hydrogen molecule, and in presence of molecular oxygen and enzyme cytochrome oxidase, it get reduced to a purple colored compound, and this oxidized cytochrome C in the presence of a reagent, that is a para amino dimethylalanine oxalate, it get reduced to a blue colored compound. The, the oxidase test detects the presence of a cytochrome oxidase system that will catalyze the transport of an electron between electron donors in bacteria and a redox di tetramethyl paraphenylene diamine. The dye is reduced to a deep purple color, and this test is used for identification of the organism like Pseudomonas, Neisseria, Alkylgens, Aeromonas, Campylobacter, Vibrio, Brucella, and Pasteurella. All of these organisms produce an enzyme cytochrome oxidase. That means these all organisms are oxidase positive. The principle of oxidase test: the cytochrome containing organism produce a intracellular oxidase enzyme. This oxidase enzyme catalyzes the oxidation of cytochrome C. The cytochrome C is one of the important carrier molecule in a electron transport chain. Organisms which contains a cytochrome C as a part of their respiratory chain are oxidase positive, and this organism turns the reagent, that is a oxidase reagent, into a blue or purple color, whereas the organism lacking the cytochrome C as their respiratory chain do not oxidize the reagent and leaving it a colorless. So means after performing this test, the colorless compound is formed, the test is negative. Blue colored compound is formed, then test is positive. The oxidase positive bacteria possess the cytochrome oxidase or indophenol oxidase. These are iron containing heme proteins. Both of these catalyzes the transport of electron from donor compound, that is NADH, to the electron acceptor, usually the oxygen. In case of aerobic respiration, the test reagent, that is N tetramethyl paraphenylene diamine dihydrochloride. This is a chemical name of the oxidase dye, and this acts as an artificial electron acceptor for the enzyme oxidase. The oxidizer reagent forms colored compound, that is a indophenol, and it is a blue colored. The number of reagents are used for this test. We can make a use of a Kovacs oxidase reagent, or a Gordon and MacLeod reagent, or a Gabe and Hadley reagent. Purpose or uses of oxidase test. The oxidase test is used to determine if an organism possesses the cytochrome oxidase enzyme. The test is used as an aid for the differentiation of the species of Neisseria, Moraxella, Campylobacter, and a Fasciola species. These all are oxidase positive. It is also used to differentiate a Pseudomonas from a related species. Here some examples of oxidase positive organisms. These are Pseudomonas, Neisseria, Alcligens, Aeromonas, Campylobacter, Vibrio, Brucella, Pasturella, Moraxella, Helicobacter pylori, Legionella, and Pneumophila. Oxidase negative organisms, for example E. coli, and this test specifically is used for identification of E. coli from Enterobacteriaceae members. Procedure for this test: a strip of a filter paper which is soaked in a little freshly made 1% solution of the oxidate dye. Then the paper is dried. After drying of a paper, a culture is rubbed on it through a wire loop. The positive reaction is indicated by a formation of a deep purple color. If such color is not formed within a 5 to 10 seconds or in a 1 minute, then we can say that test is negative. So here in this image, after rubbing one colony on oxidate dye paper, purple colored formed. So the organism is oxidate positive. It produces an enzyme cytochrome oxidase. Then next, catalase test. This test is used to determine the ability of organism to produce an enzyme catalase. Certain microorganisms produce hydrogen peroxide during aerobic respiration. Such kind of hydrogen peroxide is formed by aerobic as well as anaerobic organisms. But the aerobic organism having an enzyme catalase in their cytoplasm, and hence the hydrogen peroxide formed in respiration get denatured. But the anaerobic organism, when they are growing in presence of oxygen, the oxygen become toxic for them and they lack a catalase, and the catalase can breaks up the hydrogen peroxide, and due to lack of catalase in an anaerobic organism, the hydrogen peroxide become toxic for them. Sometimes the toxic superoxide or super radicals are formed, and these radicals does not get breaked as the anaerobic organism lacks a catalase. But the aerobic organism has a catalase. They can break down a hydrogen peroxide into oxygen and hydrogen. These are extremely toxic because they are powerful oxidizing agent and destroy a cellular constituent very rapidly. A bacterium must be able to protect itself against such O2 products or it will be killed. Many bacteria possess an enzyme that afford a protection against toxic O2 products. The catalase test is used to demonstrate the presence of a catalase enzyme. This enzyme catalyzes the release of oxygen from hydrogen peroxide. It is used to differentiate those bacteria produces an enzyme catalase such as Staphylococcus from a non-catalase producing bacteria such as Streptococci. Normally, the 3% of hydrogen peroxide is used to performing this test. Principle of the catalase test. The enzyme catalase mediates the breakdown of a hydrogen peroxide into a oxygen and a water. The presence of enzyme in a bacterial isolate is evidence when a small inoculum is introduced into a hydrogen peroxide solution and there is a elaboration of oxygen bubbles. The lack of catalase is shown by a lack of a bubble formation. The bacteria thereby protects themselves from a lethal effect of a hydrogen peroxide, which is accumulated as an end product of aerobic carbohydrate metabolism. The purpose of this test: the morphologically similar enterococcus or a streptococcus. Actually, the streptococcus are catalase negative, and staphylococcus are a catalase positive. So this test is useful for a differentiation of these two organisms. It is also useful in a differentiation of aerobic and obligate anaerobic bacteria. It is used to differentiate aerotolerant strains of a Clostridium. The procedure for a catalase test, it can be done by a tube method or a slide method. The tube method, in one test tube you have to take a 3% hydrogen peroxide, and in that tube at aseptic condition by using a sterile wire loopful suspension of a test organism or a colony is immersed and observe for a formation of a bubble. If the bubbles are formed, then we can say that test is positive. Organism can produce an enzyme catalase. It can be done by using a slide method. In a slide method, by using a wire loop or a sterile wire loop, a colony of a test organism is taken on a slide. On that same slide, a drop of 3% H2O2 is kept and observe for evolution of oxygen bubbles. If we inoculate a loopful suspension or a colony into a tube in which there is a presence of hydrogen peroxide, then such kind of bubble will be formed. If such bubbles are formed after dipping a colony of a test organism, then a conclusion is that test is positive. And in a slide test, such copious bubbles are produced which shows a positive catalase test. So thank you for watching my video. Subscribe my channel Easy Life Science. Like my video, share it to your friends and press the bell icon so you will get a notification of my newly uploaded video.